Hi everyone, welcome to this Microfluidics Innovation Center video tutorial. Here we show you how to fill a recirculation bridge for continuous cell perfusion with a pressure-driven flow controller, purging all the air from the circuit. We start with the bridge ready. See our separate bridge assembly video to get to this point. Recirculation with the pressure controller uses two reservoirs, alternately flowing and collecting. Reservoirs are connected with pneumatic tubing to the pressure controller. Our flow sensor is connected to the pump with a feedback cable. Our bridge is assembled from T-junctions and check valves. The flow will first be driven from reservoir 1 into corner 4 of the bridge, as shown in the schematic, and move this way to exit at corner 1. Fluid will then move through the flow sensor, the chip, and back into the bridge at corner 3. Then move this way through to corner 2 and collect back in the second reservoir. To fill the bridge, we are first going to tilt it to have corners 1 and 2 pointing upwards to help the air escape. To get started, add some pressure to reservoir 1 using your flow controller. Watch the liquid enter the bridge and move through the tubing. Now it's arriving back to the bridge, so angle corners 1 and 2 upwards to help the air escape. At this point, liquid is slowly filling a check valve, which has a volume of about 0.5 ml. Tapping the valves or tubing can be useful at this time to help dislodge bubbles. Make sure to continue flow in this direction until the liquid front has reached the collection reservoir. All air expelled into reservoir 2 will partition with the air in the top of this falcon tube. Now the bridge is almost filled along one flow path, pushing from reservoir 1. The next step will be to purge the remaining air from the bridge by pushing liquid from reservoir 2. In this flow path, liquid will enter the bridge at this bottom corner 2 and travel up and exit from this top corner 1. Liquid will always exit the bridge from corner 1, regardless of which reservoir is being pressurized, and then come through the flow sensor and chip in the same direction as before, re-enter the bridge at corner 3 and move up and exit at corner 4. This time to purge, we will hold the bridge in this orientation so the air in the two empty valves can escape upwards, and finally rotate corner 4 up for the final purge of the bridge. Check that you have plenty of liquid in reservoir 2. Then stop pushing from reservoir 1 and start the flow from reservoir 2. Watch the liquid filling the valves between corners 1 and 2 and corners 3 and 4 and moving upwards. Tap the valves to help dislodge the air. Air will move through the rest of the circuit and enter back in at corner 3. This is why the chip with cells should not be in line at this time. You don't want air flowing over the cells. Track the final bubble as it moves through the valves between corners 3 and 4. And once it nears the T-junction, hold corner 4 upwards to make it easier to escape. You might need to alternate flow from reservoir 1, then 2, one more time if you notice a small trapped air pocket. Make sure to flow in each direction long enough that the air bubble exits the entire circuit and reaches the reservoir. Otherwise, it will stay in the tubing and re-enter the bridge when you switch to push from the other reservoir. And now your bridge is completely filled with liquid, ready to attach your chip and start your experiment. If you found this content useful, please like and subscribe to get notified of new video releases. Thanks for watching.